Hallelujah. Hallelujah. Yana cewa ina gama yau zan dafa za mu soya kaza. Da ita za yi. Ko kuma wacce ka so dai. Ɗalibai da sun yi attending lecture.
Please don't make a noise, so I will start the second session. This is the lecture of today. The ammonia metabolism is very, very important, okay? Ammonia comes from deamination of amino acids. And ammonia is very, very toxic. As I said, nitrogen is toxic. Ammonia is toxic. Ammonia is NH3, nitrogen and 3 hydrogen atom. The main source is removal of the amino group from amino acid. Other minor source, catabolism of purines, catabolism of pyrimidines. Do you know purine and pyrimidine? Please answer me. Please respond. Do you know purine
Yes.
and pyrimidine?
Okay, doctor.
Yes.
Yes.
Catabolism
Yes, we
of
know
purines
it
and
on the amino
pyrimidines have
acid.
nitrogen. Purines and pyrimidine have
Sorry?
nitrogen. So their catabolism yields ammonia. Putrefaction of the bacteria of the large intestine act on the proteins of our diet and liberate ammonia. And this occurs when there is liver diseases such as liver cirrhosis or hepatic coma. Okay? In any liver disease, the ammonia will increase in the blood because the bacteria on the intestine liberates ammonia from the protein. But the major source is deamination of amino acid. This is the main source of ammonia. What are the fate of ammonia? Ammonia has anabolic fate and catabolic fate. Anabolic means building, synthesis. Ammonia can be utilized for synthesis of non-essential amino acids. Ammonia is used for building of non-essential amino acids. Ammonia, as we will see, enter in the synthesis of glutamine. What is glutamine? It is glutamic acid, and we bind ammonia on glutamic acid. So it is converted into glutamine. We are going to study this on the diagram. So, anabolic state of ammonia, building or the synthesis of non-essential amino acid, synthesis of glutamine, and glutamine is used in the synthesis of other compounds, such as purine, pyrimidine, amino sugar. Please, I hear noise. I'm going to mute all of you. I hear noise. Please don't make noise, otherwise I'm going to mute all of you. Okay? Another catabolic or excretory phase. Ammonia is converted into urea by the liver. Ammonia is converted into urea by the liver, and each tissue had a particular pathway for removal of the ammonia. The first thing is the liver. The liver is the main organ which removes ammonia from the blood. As I said, ammonia is toxic. I should remove ammonia from the blood. The liver, I'm going to stop and to mute all of you because there is a noise. I don't know from where this noise comes. From where? Somebody makes some noise in his room by mobilizing things. So there is a great noise. I'm going to mute all of you. The brain. The brain does not have enzymes for urea synthesis. As I said, ammonia is toxic. It should be removed from the blood. It is toxic to the brain. It leads to death. Stop the brain function. It's highly toxic. The liver removes ammonia from the blood by converting ammonia into urea. But the brain does not have enzymes of urea synthesis. The brain takes ammonia and ligates the ammonia to glutamic, conjugate ammonia to glutamic, and sensitizes glutamine, okay? And then glutamine from the brain will be translocated and transported to the liver, and the liver will get rid of the ammonia of the glutamine and glutamic and convert it into urea. Again, the brain does not have enzymes of urea synthesis. So ammonia is conjugated with glutamic and converted into glutamine. Glutamic is converted to glutamine. So it will be non-toxic to the brain because it is conjugated now to glutamic. Glutamine go outside the brain and transport it to the liver, go to the liver, and the liver get rid of ammonia, deamination of the amino group of glutamine and glutamic, and convert it into urea. The kidney. The kidney has enzyme called the glutaminase. Glutaminase enzyme hydrolysis of glutamine to remove ammonia from glutamine. So 60% of ammonia which goes in the urine come from glutaminase. 40% of ammonia which are excreted in the urine comes from deamination of amino acid, as we have enzymes, oxidative deamination enzymes in the kidney. We have transamination enzymes in the kidney. The kidney catabolize amino acid and remove amino group from amino acid by oxidative deamination, transamination. Ammonia will go outside directly in the urine, or glutamine is hydrolyzed in the kidney by glutaminase enzyme and ammonia is liberated. The last thing, the muscle, we are going to study the muscle by diagram, okay? One is raising hand. What is this? Unmute all. Anyone want to ask any question before I go to the diagram?
There is network interruption, doctor.
I don't hear. What do you need?
Yeah, there is network interruption. So in explaining the case of ammonia in urine, in kidney, we didn't understand. In kidney.
Okay, I will repeat again and I will change the network I connect. Okay, I will connect to another network. فين يا ابني بتاع راحت فين؟ في ايه؟ I will repeat again. Do you hear me now?
Yeah.
Can you
Yeah,
hear
you
me? Please don't
Yes.
make noise. So please don't make noise. So I will not be obligated to mute all of you. This is an interruption. Don't make noise in your home. This is an interruption for me. I cannot explain with noise. So I will not be able to explain. Okay? So if there is interruption, you will ask me, so the internet is interrupted, so I can repeat again. If you make noise, I will be obligated to mute all of you. Okay, I'm going to repeat again now. Again, as I said, ammonia is toxic to the brain. It will lead to death and coma. So the liver has to remove ammonia by converting it into urea. The liver is the only organ in the body which have enzymes of urea synthesis. The liver is the only organ which is able to form urea. Okay, the brain does not have the enzymes which form urea. How can the brain get rid of ammonia? The brain conjugate ammonia to glutamic to make glutamine. When ammonia is conjugated to glutamic, it will be not non-toxic because glutamine is non-toxic. By glutamine synthetase, because the brain has glutamine synthetase. Okay? This is the major mechanism of removal of ammonia from the brain. Then glutamine is transported to the liver and kidney. Glutamine go outside the brain, go to the liver, and the liver remove ammonia from glutamine, convert it into urea. The kidney remove ammonia from glutamine because the kidney has glutaminase hydrolysis, hydrolytic enzyme which breaks glutamine and get rid of ammonia. Okay? Do you understand how the brain get rid of ammonia? Do you hear me? Please answer.
Yes.
Yes, please answer because I will make sure that you are hearing and there is no problem in the internet. Okay, the kidney. The kidney has glutaminase. Glutamine from the brain go also to the kidney. And the kidney has hydrolytic enzyme called glutaminase hydrolysis. It breaks glutamine and removes ammonia, and the ammonia go directly in the urine, excreted in the urine. Another mechanism, 40% of ammonia comes from deamination of amino acids in the kidney. The kidney has enzymes which catalyze deamination. The kidney has transaminases. The kidney has oxidative deamination enzyme, and it removes ammonia, and the ammonia is excreted directly in the urine. The last thing, the muscle, I'm going to explain the muscle from the diagram. Okay. We can say the muscle at first. In the muscle, all amino acids, any amino acid in the muscle undergoes transamination. What is transamination? Exchange of amino group. The amino group will be translocated or transported to ketoglutaric. Okay? All amino acids of the muscle give the amino group to ketoglutaric. And the amino acid will form keto acid. And the ketoglutaric, when it accepts the amino group, is converted into glutamic. And I said that glutamic is a king. Why he is a king or a president? Because glutamic accepts amino group from all amino acids. There is a specific enzyme in the muscle which transfer amino group from glutamic to pyruvic. Pyruvic is an organic acid. Pyruvic in the muscle gets the amino group from glutamic, but this reaction is unique to the muscle, occurs only in the muscle. Pyruvic accepts the amino group from glutamic and is converted to alanine. Alanine is another amino acid. So these are actually two reactions, not single reaction. So this is a muscle cell. This is a muscle. Okay? Again, all amino acids, I will make an arrow around it, gives the amino group to alpha ketoglutaric. And the amino group, when it is conjugated to alpha ketoglutaric, it is converted to glutamic acid. This is an amino acid. Again, the amino group will make another journey. The amino group will be transferred to pyruvic. When pyruvic gets the amino group from glutamic, it is converted to alanine. And then the muscle liberates alanine to the systemic circulation, and the alanine go to the liver. In the liver has enzymes which catalyzes removal of the amino group from alanine by transamination, oxidative deamination, and so on. And amino group is liberated in the form of ammonia, and the liver has enzymes which sensitizes urea. Urea is non-toxic, go to the blood, go to the kidney to be excreted in the urine. Okay? Of course, alanine, when I get the amino group from alanine, it is converted to pyruvic. Pyruvic can be converted into glucose. Okay? By gluconeogenesis. If you study carbohydrate metabolism, you will know that pyruvic can give me glucose by a pathway called gluconeogenesis. Here there is a cycle. Glucose will be liberated to the systemic circulation and make a great journey and return to the muscle to be oxidized by glycolysis to give energy and to give me pyruvic. This is a spelling mistake. This is R, not T. Pyruvic acid. So this is called glucose-alanine cycle. Everyone understand the glucose-alanine cycle, or it is not clear?
We understand, doctor.
Okay, I need another person to say that he understands the glucose-alanine cycle.
Yeah, I understood it, but you return me the slide, doctor. I want to screenshot it.
I will share the slide again, and I will publish the video on the YouTube channel to see it again. Okay.
Right.
I will repeat the slide again. Again, these are the muscle cells. I will make here by a yellow color highlighter. These are the muscle cells. If you see this muscle cell have a hexagonal or has 8 sides, these are the muscle cells. Inside the muscle cells, we have all amino acids. These are amino acids. 20 or 30 amino acids. As I said, we have more than 20. The 20 are amino acids of the protein, but we have another amino acid not found in the protein. All amino acids will give the amino group to the king. Who is the king? Who is the king? I said, who is the king? The king is glutamic, because the keto acid of glutamic, alpha ketoglutaric, takes the amino group from all amino acids. And the ketoglutaric is converted into glutamic, okay? This process is called transaminase, exchange of the amino group. Glutamic is a king because it accepts the amino group from all amino acids. Again, there is a unique reaction to the muscle, ALT. Pyruvic acid is an organic acid. The amino group of the glutamic will make another journey. It will be transferred to pyruvic. Pyruvic will accept the amino group of glutamic, converted to alanine. Alanine will make a third journey. It will go to the bloodstream, go to the liver. This is the liver cell, okay? Alanine, by deamination, it will give amino group, and the liver has enzymes which sensitizes urea. Ammonia is toxic. NH3 is toxic, but urea is non-toxic. Urea go to the bloodstream, go to the kidney to be excreted in the urine. Alanine, when we remove amino group of alanine, it is converted to pyruvic. If you study carbohydrate metabolism, pyruvic can give me glucose by a pathway called the gluconeogenesis. And then glucose will make a great journey, go to the bloodstream, and then return to the muscle. Glucose is oxidized by glycolysis, and it will give pyruvic acid. Okay? I will stop. This is called glucose-alanine cycle. Is the glucose-alanine cycle clear? Please answer.
Yes, it is clear, doctor.
Okay. If anyone want to ask any question, please write it in the chat box. Okay? I will share again. Urea cycle is very important, and there will be many questions in the exam about urea cycle, because as I said, ammonia is highly toxic to the brain. Ammonia is converted into urea by the liver to remove the toxicity of ammonia. Okay? Urea is excreted in the urine. I will make the pen red color, red pen. Okay, where does urea cycle occur? Urea cycle occurs in the liver only in the liver. Why? Because enzymes of urea cycle are found only in the liver. No other organ in the body. No other organ in the body can sensitize urea. The liver is the only organ which sensitizes urea. Where does urea occur? Occur in the mitochondria and in the cytosol. We have 2 reactions in the mitochondria and many reactions in the cytosol. So if you are asked in the exam, where does urea cycle occur? It occurs in the mitochondria and in the cytosol. It occurs only in the liver. Okay?
Yes.
This is an example. Arginase enzyme is found only in the liver. This is the last enzyme which sensitizes urea. It is found only in the liver. It's very important to know the overall reaction. Yani the overall reaction. Because we have several reactions, about 7 reactions in the urea cycle. It is a pathway. But the overall, what does occur? We consume 3 adenosine triphosphate. 2 of them will be broken down into adenosine di. 1 of the 3 will be broken into adenosine mono. We consume 3 adenosine triphosphate. 2 of them will give me 2 adenosine di and 2 phosphoric acid. 1 of the 3 molecule will be broken into mono, adenosine mono, and PPI. PPI means 2 phosphoric acid conjugated together. They are called pyrophosphate. We begin with what? With carbon dioxide. This is a use of carbon dioxide. Carbon dioxide, which comes from oxidation of glucose in glycolysis and citric acid cycle. So this is one of the uses of carbon dioxide, synthesis of urea. Ammonia, which comes from deamination of amino acids. As I said, ammonia is toxic. Ammonia will be conjugated to carbon dioxide. Another amino group, we get it from aspartic. So very important, aspartic. Aspartic gives me a second amino group. Actually, I need 2 amino groups to sensitize urea. As you will see, urea has 2 amino groups. One of them is a free amino group. The second amino group comes from aspartic. I need also carbon dioxide to sensitize urea and fumaric acid. We are going to discuss the details of urea cycle enzymes. Do you see this blue area? This blue area is the mitochondria. These are the mitochondria. I have 2 enzymes which occur in the mitochondria. And what is this? What is this area? What is this area? What is this? Anyone can tell me what is this? What are these circles?
This matrix.
Huh?
This matrix of
No. What are these circles? This circle, the yellow circle. And we have another line of a yellow circle. What are these circles?
Phospholipids.
Phospholipids.
What are these? Anyone? I discussed this in the electron transported chain for those who attend the electron transported chain with me. This is the mitochondrial membrane, the inner mitochondrial membrane. This circle is the phospholipids. phospholipid, the membrane of phospholipids. And this is another layer of phospholipid. We have two layers of phospholipid. This is the mitochondrial membrane. And all this area is the mitochondrial matrix. I am now inside the mitochondria. This blue area is the mitochondrial matrix. As you see, it is written here, this is the mitochondria. Okay? And the yellow color is the membrane. So below this yellow color, I am in the cytosol. Do you understand?
Okay.
You understand? We are now inside the mitochondria. This blue area, the light blue area, is the mitochondria matrix. And this yellow color is the membrane. This is the membrane. I go with the mouse. This is the membrane. It has two layers of phospholipid, the mitochondrial membrane. The first enzyme, very, very important, is called CPS1. CPS1. What is CPS? Carbamoyl phosphate synthetase 1. Carbamoyl phosphate synthetase 1. Carbamoyl, capital C. Phosphate, capital P. Synthetase, capital S. Number 1, this is the first enzyme because I have number 2. Because I have carbamoyl phosphate synthetase 2. Okay? Here in the urea, I don't have 2. I have carbamoyl phosphate synthetase 1. I should have ammonia because this is the toxic compound we need to remove. I said before that these are uses of carbon dioxide which comes from oxidation of glucose. I will conjugate ammonia to carbon dioxide. Any conjugation of ammonia will remove the toxicity of ammonia, okay? When I conjugate ammonia to any molecule, the toxicity decreases. I conjugate ammonia to carbon dioxide. For any conjugation, I need energy. Comes from adenosine triphosphate because I make covalent bond. I create covalent bond. I need another molecule of adenosine triphosphate, so I need in this reaction 2 molecules of adenosine triphosphate. What will do? The ammonia will be conjugated to carbon dioxide, COO. These are carbon dioxide. And there is also phosphoric acid. Phosphoric acid is conjugated by high energy bond. As you see, this is the high energy bond. We are going to go outside the meeting within 10 minutes and to return again. This Zoom meeting will end within 10 minutes, okay?
All right.
Again, CPS1 come in the exam by abbreviation. So you have to know the abbreviation. CPS1, very important. The name of the enzymes of urea cycle. Because there are many diseases in the urea cycle. Carbamoyl phosphate synthetase 1. Conjugate ammonia to carbon dioxide. I need 2 molecules of adenosine triphosphate. This is the first, this is the second. This is ammonia, and this is carbon dioxide. This is called carbamoic acid. Carbamoic acid conjugated to phosphoric acid, but this high energy bond. This is called carbamoyl phosphate. What is this? This is an activator. As you see, some enzymes needs activator. Certain enzymes needs magnesium. Other enzymes needs calcium. Some enzymes needs hormones to be activated. This enzyme needs N-acetyl-glutamic. Glutamic acid with acetic acid attached to it, called N-acetyl-glutamate. Carbamoyl phosphate. This reaction occurs in the mitochondria. We'll go to a second reaction, which is called ornithine transcarbamylase. You have to know the name of the enzyme. Very important. Ornithine will give me a second amino group. So urea actually has two amino group. This is amino group and this is amino group. So the second amino group comes from ornithine, but we will see it comes from aspartic, as you will see. Ornithine is an amino acid which is not found in protein. As I say before, we have many amino acids in our body which is not found in protein. We are free amino acids. Ornithine will be conjugated to carbamoyl phosphate. What is R? I said R in the first part of the lecture. What is R? R is, anyone remember what is R? Do you hear me? Please answer me. Do you hear?
No, I don't remember R, doctor.
Do you remember what's R?
No.
R is a hydrocarbon, the tail of ornithine, the remaining of the structure of ornithine, hydrogen and carbon. The most important is the amino group of ornithine. It will come here. So this is the beginning of building of urea. Urea has two amino group and ketone, C-double-bond-O. This amino acid is called citrulline. Again, citrulline is amino acid found in our body but does not enter in the structure of the protein. So we have more than 20 amino acids in our body. The 20 amino acids are those which enter in the structure of protein. But we have another amino acid which does not enter in the structure of protein, but play a very important role in metabolism. Citrulline will cross the mitochondrial membrane. I said that mitochondrial membrane looks like the castle or the home of the king, okay? The house of the king. It's not easy to cross the border or the wall of the castle of the king, okay? This is a king, the mitochondrial membrane. As I said, this is a layer of phospholipid. This is another layer of phospholipid. Mitochondrial membrane is written in yellow. Citrulline has to go to cross the mitochondrial membrane to go outside to the cytosol. So the first two reactions occur in the mitochondria, okay? This is a transporter, which allows citrulline to cross the membrane and go from the matrix outside to go to the cytosol in exchange for ornithine. Ornithine will come from the cytosol and go enter the mitochondrial matrix. So citrulline will go out of the mitochondrial matrix and ornithine will enter. This process occurs simultaneously at the same time, so it is called antiport. Antiport means two molecules runs in opposite direction. Citrulline will go outside the mitochondria. Ornithine will cross and enter the mitochondria. Okay, we are going to the second slide. This is the mitochondrial membrane. Citrulline, this is citrulline. Citrulline will go. It will react with aspartic. As I said, aspartic play very important role, okay? You don't have to know the formula, okay? You don't have to know the formula. But I want to remind you of something. The name of the enzyme is very important. If you don't want to study the cycle, you must know the name of the enzyme. The third enzyme, we are now in the cytosol. We go outside the mitochondria. Inside the mitochondria is a blue color. Outside the mitochondria is a yellow color. The third enzyme is in the cytosol. Its name is arginine succinic synthetase. Arginine is an amino acid. Succinic is an organic acid. Synthetase means I conjugate arginine with succinic. Here is adenosine triphosphate is broken into mono. And 2-phosphoric acid will be liberated. Aspartic acid is an amino acid. Aspartic has an amino group. I want you to notice something. What is the color of aspartic? Aspartic is written in which color?
Blue.
Blue. Citrulline is written in a dark blue. I want to go to the citrulline. I will go to the previous. Okay. If you look at the citrulline, it is written in a dark blue. Dark blue. Aspartic in a light blue. This is the citrulline in a dark blue. You notice this is amino group. Comes from citrulline. Amino group comes from citrulline. And this is an R of citrulline. If you look here, a third amino group. A third amino group. So we have 3 amino group here. This one comes from aspartic. Okay? This one comes from aspartic. How do I know that it comes from aspartic? From the color. The color is light blue. The color here is light blue. And this is the remainder of aspartic acid. The remainder of aspartic. Okay? So I have carbon atom attached to 3 amino group. Actually, it is amino, not amino. NH, NH, NH. This group is called guanido. Guanido. Okay? I have 3 amino group. But as I said, urea is formed of 2 amino group only. The first amino group comes from ammonia. As you see, the first amino group from ammonia. The second does not come from citrulline nor ornithine. The second amino group comes from aspartic. If you look at urea, this is the second amino group which comes from aspartic. The first amino group comes from ammonia, okay? This structure is called arginine succinic. Of course, you don't have to know the formula of arginine. You don't have to know the formula of succinic, but you have to know the name of the enzyme. Argininosuccinate synthetase, and this enzyme breaks adenosine triphosphate into adenosine mono. This is very complicated structure because it is formed of 2 amino acids, of 2 organic acids. Arginine is an amino acid, succinic is an organic acid. An enzyme which is called lyase or hydrolase enzyme. Argininosuccinate ase. Ase means hydrolysis or breaking. We break here. If you find there is a dash here, we break here. If we break here, we get fumaric. And this is the remainder of the structure. Arginine, arginine, arginine has guanidine group. Arginine has first nitrogen, second nitrogen, third nitrogen. So this is a guanido group. Arginine is broken by hydrolysis. Arginase. Ase means hydrolysis to liberate urea. If you look at urea, the first amino group comes from aspartic, which is blue color. The second amino